In 1962, two men stood on a stage in Stockholm and accepted science's highest honor for a discovery they didn't actually make. The real work had already been done by someone else, someone who wasn't in that room, someone who, by then, was already dead. This is the story of Rosalind Franklin and the photograph that changed biology forever. By the early 1950s, scientists knew DNA carried genetic information. What they didn't know was its shape. And shape, in molecular biology, is everything. Get the structure right, and you understand how life copies itself. Get it wrong, and you're stuck. Labs across Britain and America were racing to solve it. At Cambridge, two researchers, James Watson, a young American, and Francis Crick, a British physicist turned biologist, were building physical models, twisting wire and cardboard, trying to guess their way to the answer. But guessing wasn't going to be enough. Someone needed to actually see the structure, and that required mastery of one of the most brutally difficult techniques in science, X-ray crystallography, and that person was none other than Rosalind Franklin. Franklin was a physical chemist working at King's College London, and she was, by any measure, one of the best X-ray crystallographers in the world. The technique she used involved firing X-rays at fibers of crystallized DNA and capturing the way they scattered on photographic film, a diffraction pattern. From the pattern of Lysian shadow, an expert could mathematically reconstruct the molecule's three-dimensional shape. It was slow, exacting work. It required DNA samples, humidified to a precise degree, exposure times lasting days, and the mathematical skill to interpret patterns that looked to anyone else like meaningless smudges. Franklin was meticulous. She refused to publish guesses. She wanted certainty. In May 1952, after months of refining her technique, she produced an X-ray photograph of DNA in its hydrated B form. It would later be called the most beautiful X-ray photograph of any substance ever taken. It was labeled simply Photo 51. Photo 51 showed a clear X-shaped pattern, and to a trained eye, an X shape in this kind of diffraction image is the unmistakable signature of a helix. Franklin's own unpublished notes show she had already deduced the backbone's helical structure and worked out key structural measurements. She was close. She wanted to confirm her calculations before publishing. But at King's, Franklin worked in a tense environment. She was one of the only female scientists in her department, often left out of contemporary academic socializing and treated with a degree of friction by colleagues, including a fellow researcher named Maurice Wilkins, who also worked on DNA at the same lab. In early 1953, without Franklin's knowledge, Wilkins showed Photo 51 to James Watson. The effect was immediate. Watson recognized the X pattern instantly, and combined with a separately leaked summary of Franklin's unpublished calculations supplied through an internal Research Council report, he and Crick now had the missing structural data they'd been unable to solve on their own. Within weeks, using Franklin's evidence to validate the geometry, Watson and Crick built their famous model, the double helix, two strands twisted around each other like a spiral staircase connected by base pairs. They published in Nature in April 1953. Franklin's data was acknowledged only briefly in supporting papers published alongside theirs, framed as if it merely confirmed a structure Watson and Crick had already reasoned out independently. The full story of how her unpublished photograph and analysis had actually reached them stayed hidden for years. Franklin left King's College not long after, moving to Birkbeck College, where she shifted her crystallography skills toward the study of viruses, work that was, in its own right, groundbreaking. She published over a dozen papers in just a few years there. In 1956, she was diagnosed with ovarian cancer. Some historians have pointed to her prolonged occupational exposure to X-ray radiation as a plausible contributing factor, though this has never been definitively established. She continued working through her illness, publishing research from her hospital bed. She died in 1958. She was 37 years old. 4 years later, Watson, Crick, and Wilkins received the Nobel Prize in Physiology or Medicine for the discovery of DNA's structure. The Nobel Committee does not award prizes posthumously, so even if attitudes had been different, Franklin's death had already written her out of contention. It wasn't until Watson published his own memoir years later, one that described Franklin in dismissive, often personal terms, that public attention turned back toward her actual contribution. Colleagues who had known her pushed back hard, and historians of science began re-examining the lab notebooks, correspondence, and timeline in detail. What they found was clear. Franklin's data wasn't a footnote to the discovery of the double helix. It was the missing piece that made the discovery possible at all. Today, her photograph, Photo 51, is recognized as one of the most important images in the history of science. Institutes, scholarships, and even a Mars rover have been named in her honor. Rosalind Franklin never set out to be a symbol. She wanted to be a scientist, rigorous, careful, certain, before she spoke. In the end, the universe didn't need her to be remembered to be right. The helix was always there in her data, written in X-ray light, whether anyone gave her credit or not. But we get to choose what we remember, and maybe it's time her name came first.